Record. All right. So today, for our second class of review, we'll be looking at enzymes, ATP, cellular respiration, photosynthesis. And I've got new questions for you guys. And so I'm actually going to start with a question. So if you want to, if you're at home, you can access these pretty easily. Let me show you really fast where stuff is at. Oh, whoops, hang on, I'm in student view, so it won't let me look. Leave student view. All right, so if you're at home and you want access to these, I've got this one file that has all the free response that will potentially, we won't get to all of them, I always kind of overpack. But I've got a file with all the free response that we'll be looking at for like the rest of the year. So instead of having to like dig them up, it's just one PDF file. So I'm going to show you guys where that's at. So if I go to our Google Drive address is that. Oh, thanks, Kim. I go to AP Bio, and I've got it in the notes section. So AP Bio notes. And then I just called it Unit 8, just because you'd probably be looking through for like the newest looking thing. So Unit 8 says AP Bio Review Free Response and Ultimate Reviews. So if I click on that, then these are the free response that we'll use when you're in the after school sections and the after school sessions. And I pull up an old review question, an old free response, it comes from here. When we're in class and we're reviewing, I'm pulling it from here. So these are all the free response that we might potentially do between during class and the after school sessions. So if I click on that, then I'm good to go. Come on, Google Drive. It's not a video. There we go. All right. So in after school review yesterday, we did 2010 number 1. Here's 2010 number 2. Now, I'm going to get out of Google Docs and use my own saved PDF version. But here you go. All right. Now, on the real test, they had like a little data table, like some grid paper that you would graph it on. You don't have to sit down and like measure everything. Just do like a real quick, rough kind of, here's the graph, here's sort of what it would look like type of thing for part A. So here's what it says. An experiment was conducted to measure the reaction rate of human salivary enzyme alpha amylase. Amylase is the stuff that breaks down starch into sugar. So like if you eat crackers or bread, it starts breaking down into sugars in your mouth because of amylase. 10 milliliters of a concentrated starch solution and 1 ml of alpha amylase solution were placed in a test tube. Okay, starch is my substrate. I added in with the enzyme amylase. The test tube was inverted several times to mix it, incubated at 25 Celsius. So, you know, it's a little bit warmer than room temperature. That's probably about 80. The amount of product, maltose, present was measured every 10 minutes for an hour. And here's my data. Okay, so we are eventually going to answer like all 4 of these, A, B, C, and D. I'll kind of go through them one at a time, but if you get ahead of me, you're welcome to move on ahead. So part A says, graph the data on the axes provided and calculate the rate of reaction for the time period 0 to 30 minutes. So give it a shot. And again, you can just hand draw a graph. You don't have to get all picky. You just sort of, there you go, and give me a rough guide of what the graph might look like, and then you can calculate the rate. The likelihood that they have you calculate the rate of something is almost a guarantee. shrink my picture just a little bit here. Graph the data on the axes provided. And again, they gave you like a piece of kind of grid paper. And calculate the rate of the reaction for the time period 0 to 30 minutes. And for in-person, did I get everybody's quizzes up here? Okay, now, as we're doing this, I've got like on our general, just like our syllabus, I kind of tell you what which ones of these are coming up, like which ones we're going to do in class. So you can always do them ahead of time. If I end up kind of moving on too quick, you can always look ahead. Like if you look at our syllabus before review day 3, it'll be like, these are the 2 that you can work on. If you want to look at 2010 number 2, you can do it. So you can know ahead of time if you want to take your time more. All right. So I do either remember or I go, and if I want to remind myself, what did they give you credit for and not give you credit for, I look that stuff up. So you'll have to just kind of trust me. Hopefully by now you trust me. And other times, if it seems unbelievable, I'll actually pull up the rubric and it'll specifically say, like, we count this, we don't count this, so that you can be like, man, they were tough on that one, or they were super lenient on that one. Okay, so graph the data. Anytime they have you graph the data, they always have 1 or 2 points available for just what I call 7th grade graphing. So make sure, you might not have it on yours because you're just kind of hacking it together really fast, but don't forget to put a title. Don't forget, let me switch over here. So don't forget to put a title of some kind. In general, as long as it's sort of on topic, they aren't picky. So you don't have to say like, this versus this. If you just said like enzyme experiment, they'd be like, cool. If you said US presidents, they'd be like, no, that's wrong. So a title. Don't forget to put a label and units. Maltose concentration is a label. Micromoles. No, that's not micro. Yes, that's micromoles. Micromoles is a unit. Time is a label. Minutes is a unit. So I'd have time down here. Then I'd have like min. I'd have maltose concentration. And then micromoles. So doing that, and then whenever you graph, the way that we know if your graph is big enough is if I could have doubled it and fit it on there, then it's too small. So what that means is your graph should always take up more than half of the space provided. If we gave you a graph this big and you drew this, they're going to say it's too small. They're going to say, oh, you counted by ones, but it's so small. You should have counted by halves. And now it would look like, you know, this. Oh, there, that took up less than half the height. That's too small. So you should have made your increments different so that now it took up more than half the height. So you should always be taking up more than half of the graph and more than half of the graph. Does that make sense? That's how they decide. Like otherwise, if I sit down and I grade 4,000 of these, when readers read these, you get like assigned a question and you grade it for 8 days. So like 8 hours a day, 8 days, you grade question 2. You only grade question 2. You grade hundreds of question 2s every day. So by the end, you have to be like super fair. Somebody's going to have a graph that's this big, somebody this one, somebody this, this, this. And at some point, you have to be like, here's the official cutoff for is it big enough? And so that's what they do. If I could have doubled it, then it's too small. So between all that crap that I just set up there, there's usually about 2 points available. All right, so now my graph looks like it's pretty steep slope for a while. And then somewhere around 30 minutes, it's starting to slow down a little bit, but somewhere around 30 minutes, it slows down a lot. Then it's close to flat line. So it's going to look something like this, more or less. On the real ones, you'd put hash marks, you'd have numbers and all that. So after all that crap, there's 2 points available, probably, maybe 3, but probably 2. Back in the day on this one, I think it was 3 points. And it's 3 points once we throw in the calculate. So how do I calculate the rate of the reaction for time period 0 to 30 minutes? Like, how do I set that up? What do I do? Huh? Yeah, just the slope. So change in y over change in x, or y2 minus y1 over x2 minus x1, or rise over run. And since I just sort of like ghettoed my graph right here, I'm going to use the numbers off of my data table because they're perfect, right? It's not dependent on like my line. So it says at time 0, I had a concentration of 0. That makes sense. At time 30 minutes, I had a concentration of 10.4. So change in y over change in x. 10.4 minus 0 over my time of 30 minus 0. And so that's going to give me somewhere around like a little bit about a third. So 30 something percent. What's it come out to be? 30. Anybody actually calculate it? Okay, but you set it up, right? Okay, so this is going to end up being like 0.3 something. And don't forget to put your units. It'd be micromoles per minute. All right, that's part A. In total, probably a couple points here and 1 point here. You guys are going to take your AP Bio test on paper. Same thing with you guys at home. You have to come up to school and take it on paper. So you can show work. All right, part B says explain why a change occurred in the reaction rate after 30 minutes. So my rate was somewhat constant for a while. Then it slows down some, and now it's like pretty much a flat line. So why is that? What do you think? Why did the reaction rate change after half an hour? Move that there. Huh? Yeah, we kind of ran out of starch. We used it all. So if I start with 50 pounds of sugar and I'm making cakes, eventually I run out of sugar and like, no more cakes. So sometimes students will be like, oh, denatured. And you're like, but what evidence do you have that something denatured it? Like we didn't talk about temperature or pH or anything like that. And so what that means is basically I ran out of substrate. I used up all the starch that I had. And that was the right answer. So explain why? Well, you used up all your substrate. And so now the rate is pretty much flat, pretty much 0. Okay, part C. I'll give you a second on this one. It says draw and label another line on the graph to predict the results if the concentration of amylase was doubled. If I double the concentration of amylase and I draw a new line, when I draw a new line, I'd want to make sure I label it. And whatever you label it, remember, when you're writing essays and you're like, should I write it this way or that way? Think of it as if you're writing an essay for an AP Bio teacher that's at a different school. So, for example, with me, you can't just be like, oh, it's kind of like that analogy of the airport. Whoever's reading it's going to be like, I don't know what the hell you're talking about. Like, so write it so that a professor at some college will look at it and be like certain that you know what you're talking about. So if you label this and you call this original and you call the other one, you know, 2x amylase, I, as a reader, will very easily know what the heck you're talking about. Or if you label another line and you say part C, I will know what that is. Like it's very obvious. Any idiot could be like, oh, This is part A, and that one that says 2 times amylase is the one when we doubled the amylase. All right. So what do you think? What's going to look different on my new line? Yeah, good. It's going to be steeper at first. Now, I agree. It will be steeper at first. When they graded it, they basically said if it was steeper than your original, it's good. They don't care if you like went this much or this much or this much. They just wanted it to be steeper. Where does it end? Uh-huh. Do it. Okay, so it should flatline sooner. That's true. So instead of flatlining way out here, it's going to flatline sooner. Is it going to flatline here, lower, at the same spot, or higher? Like on the y-axis. I agree, Aiden. On the x-axis, I'll get to the flat line quicker. Okay, so maybe like this. So it could be this. We'll call this option A. We'll call this option B. We'll call that option C. So A or B or C. How many of y'all think it's A? How many of y'all think it's B? How about C? It's B. What's wrong with A? Was your hand up, Aiden? You're kind of. You can give it a shot if you want. Yeah, in the original graph, I've only got like a certain amount of like substrate, right? So think of it this way. I'm making cakes. My substrate is sugar. I've got 50 pounds. In my original one, it takes me like 30 minutes and I start running out of sugar. Now I have 2 people making cakes. I've still only got 50 pounds of sugar. So I'm still going to end up with the same amount of cakes. I'm just going to be done faster. So here, I'm still going to level out eventually at the same original spot because the only thing that changed was my amount of amylase. So I just get there faster. The reaction rate is quicker, but I still only have the same amount of substrate. So going up there is not correct. This is kind of what separates like 4s and 5s on the AP Bio test. The students will be cruising along, everything's going along great, and then all of a sudden, one little thing like this here and there, and that's kind of what separates 4s and 5s. It's just like that little bitty thing where you're like, oh, well, crap, why didn't I think of that? All right, and then the last one, identify 2 environmental factors that can change the rate of enzyme-mediated reactions. Discuss how each of these 2 factors would affect the reaction rate of an enzyme. So I'll give you just a second to see if you can identify 2. In class, we've technically learned 3, but we've emphasized 2 a bunch. So I imagine that all of my students will, if they come up with 2, they'll come up with the same 2. There's technically a third one. Okay, for you guys in the chat, you can get in the mix also. It's early in the morning. Some factors. As far as the discussion part, don't sweat it. That might be too much to write. Yep, you got it, Isaac. So 2 factors that can change the rate of enzymatic reactions. All right, what do you guys think? Somebody give me one. Hmm? Yep, could be an inhibitor. Now, an environmental factor. So I agree an inhibitor could slow it down, but that might not be something that's just out in the environment. I do agree there could be some chemical, but inhibitors are generally not considered environmental. So, but I do agree, otherwise that would slow it down. Temperature. So temperature and what's the other one? pH. So temperature and pH. Those are the ones that we talk about the most. There's a third one, ion concentration. So it works the same way as pH does. So let me kind of show you what I mean. If I've got my enzyme right here, it's this chain of amino acids all folded up. And we kind of mentioned this earlier when we were going over the quiz. We were talking about acids and bases and stuff like that. So if the pH is too extreme, then a lot of times if the pH is acidic, I'll have a bunch of H pluses floating around. And so if on here, I have some areas with negatives, those H pluses can start sticking to those negatives. And when that happens, now the most stable shape of this protein might be different. So here's my protein. It's got a stable shape. Maybe right now, that and that are attracted by positives and negatives. Once I go in and start like covering up those negatives, now it changes shape. Now that enzyme doesn't work anymore. So if it's a reversible change, we call it a conformational change. If it changes it forever, we tend to call it denaturing. So too much H plus, too extreme of a pH could damage that. Same thing could happen with salt or anything else. If I add in a whole bunch of Na plus, they could also be attracted to those minuses. So just general ion concentrations, any ions, doesn't have to be acids and bases. And then temperature, how was it that temperature denatured my protein? Like, why is it logical that, oh yeah, here's what happens with temperature?
If it gets too high, it denatures the enzymes, kind of like when you fry an egg.
Yeah, kind of like when I fry an egg, when the temperature gets too high, then I like change the chemical properties. What's happening at like the molecular level? Yeah, the hydrogen bonds break. You might remember I have primary structure, or right, a correct order of a chain of amino acids. That can then fold into like alpha helices and beta pleated sheets. Those alpha helices and beta pleated sheets are held together by hydrogen bonds, like this. So what's holding the shape of these together? Hydrogen bonds. But hydrogen bonds are the weakest bonds inside of a human body. And so out on the shelf, actually out on the shelf, hydrogen bonds are the weakest also. But inside the human body, hydrogen bonds are the weakest. So whenever I heat up anything, it starts to move more. And so this little coil right here, let's say it's all coiled together, at body temperature, 98 or 99, it's kind of vibrating around like this. But as my temperature gets to 100, 102, 104, 108, something like that, then now, eventually, I get to the breaking point of hydrogen bonds, and those hydrogen bonds break. And all of a sudden, my protein like unravels. It just became denatured. So that's how temperature can have an impact. So that's what I would say when I'm discussing how each of those would affect the reaction rate. All right. Feeling okay on that question? Exciting stuff. So here's kind of the way you have to look at review. Here's how I have to look at it. As a teacher, reteaching stuff that you guys have already learned is less exciting. Like the more exciting thing would be to like pick another body system, like learn how the immune system works. That's new information. That's more fascinating than rehashing old stuff. But the way that you have to get joy out of review is when you know stuff, when all of a sudden you're like, oh, sweet, like I'm in good shape. So it's kind of like when we start talking about something and you're like, I know it, I know it. Oh, wait, I almost know it. We review it. You're like, okay, now I've got it. The way that you have to find joy in review is when you're like, okay, now I'm going to be ready for like the AP test. Or if you don't take the AP test, now I'll be ready for college biology. But something like 91% of you guys signed up for the AP bio test. So most of you guys are wanting to do well on it. So whenever we're doing this and you're like, man, I wish we could learn something new. Me too. But it is kind of cool when you remember this stuff. So think of it that way. Okay. So now what we want to do is look at a few more general things on enzymes that aren't in a question. Then I'm going to throw another question at you. So think about inhibitors. I think it was Aiden earlier that mentioned inhibitors. If I just said, hey, there's a question about inhibitors, tell me what you know about them, what would you write? Jot down just a few things that you know about inhibitors. Hey, Mr. Cotton, they asked a 3 or a 4 point question on enzyme inhibitors. What do you think earns points? That's all you know. They asked a question about inhibitors. What kind of things would they give me points for? And even if you're at home eating some cinnamon toast, that's what I'd be doing right now. I'd be eating cinnamon toast while I was in biology class because that just sounds good. Unless you have legit cinnamon rolls, then that's even better. But cinnamon toast will work in a pinch. So while I'm eating my cinnamon toast, I'm still going to make myself jot stuff down because if not, my brain kind of wanders. So still jot stuff down, even though I don't make you turn it in or anything. Yes, I agree, Isaac. Yep, I give you maybe 30 more seconds. And also, I'll show you the official directions, but you can use pictures on your essay. So the directions say something like, um, diagrams alone are insufficient for credit, but if they're labeled and explained or whatever, then you can get credit. I don't remember the exact wording, but like if you just barf up a diagram, they'll be like, no. But if the diagram helps to explain something, like if I'm talking about this and I'm writing an essay and I kind of show that, anything that helps the reader understand is fair game. That's actually why I requested that our test be only paper, because so many things are better explained with diagrams. If you're on a computer and they're like, explain transcription and translation, and you're like, oh, crap, all I've got is words. Explain how ATP synthase works, but don't draw any pictures. That sucks. All right. So how many different types of inhibitors have we learned about? Two. Does anybody remember what they were called? Huh? Yep, that's one way of classifying. That's correct. So they're called competitive and non-competitive inhibitors. So competitive and non-competitive. All right, so let's say I've got an enzyme right here, and I've got supposed to be an identical enzyme right there. Not at all. That's supposed to be the same. What's different about how does a competitive inhibitor work versus how does a non-competitive inhibitor work? Yeah. Yeah, good. With the non-competitive one, and obviously the reason I repeat is so people at home can hear. With the non-competitive ones, my inhibitor binds to a not active site. What's that spot called that's not an active site? They have a name for it. It also starts with A, but it's not active site. Good, allosteric. Thanks, Kevin. So an allosteric site. So right here, this maybe is where my substrate goes. This is the enzyme. And so an allosteric site, allo means different or other. So maybe over here on my enzyme, I've got an allosteric site. And whenever this inhibitor, whenever that inhibitor hits that allosteric site, then it turns the enzyme off. Well, how does an inhibitor hitting the enzyme way over here, far away from the active site, how does that turn this enzyme off? It looks like the active site's still good to go. Yeah, it changes the shape of the whole enzyme. So whenever I bind an inhibitor over here, now the most stable shape of this protein might be different. And so now maybe my enzyme changes shape and looks like this. So now it's off. So whenever that inhibitor hits my allosteric site, allosteric, it's one word, I separated it like it was two words, but it's one word, allosteric site. Then now what happens is my enzyme will do something like this. The active site will change. So now my substrate doesn't fit. Competitive inhibitors. What's different about a competitive inhibitor? Yeah,
It goes to the active site.
yeah, it goes to the active site. It's competing for the same spot as the substrate. So here's my substrate that wants to go in there, but then here's the inhibitor. The inhibitor will have a pretty similar shape on the bottom. You can tell why I'm not an artist. Actually, that's backwards. So the bottom will be similar, but the top might be different. And it's competing for the exact same spot. It's competing for the active site. So they call that a competitive inhibitor. The nice thing about inhibitors is when I put them in there, the enzyme's on, I'm sorry, off, and when I remove them, the enzyme goes back to on. So inhibitors usually are not permanently turning them off. Some inhibitors do. Some inhibitors do go, like land in the active site, get stuck, and never leave. So it is possible that inhibitors can be permanent. But usually in an organism, they're versatile. We can add them and remove them. Inhibitors. All right. Take maybe a minute and a half and write down what you can remember about the levels of protein structure. Levels of protein structure. Levels of protein structure. If you're totally stuck, I'll give you a hint. There are 4 levels of protein structure. Good. What you've got in there is good so far. All right. So we've got primary structure. They were very picky about how you stated this. So what was primary structure? What were they picky about? Yeah, yeah, I can. Yeah, a string of amino acids in the correct order. Or if you said somehow that you let me know that it's not just a chain of amino acids or a string of amino acids, but in the right order or the correct sequence of amino acids or the correct amino acids, something like that in sequence. Yes, you got it, Davey. Um, and so that's what they're looking for. And if I was drawing it, I might have like amino acid number 1, number 2, number 3, number 4, and so forth. Just so you know, proteins can be really big, really small, but just to give you kind of an idea, most of them are a few hundred amino acids long. Some are thousands. Some of the really big ones are tens of thousands. But just to kind of give you an idea, like most proteins aren't like 20 amino acids long. They're usually 100 and something amino acids long. Okay, secondary structure. I had two shapes that pop up. What were the two shapes? Yeah. Yep, alpha helix and beta pleated sheet. I even had some drawn over here on the board a little while ago. I've erased them, obviously. But I take the same string and I start kind of making these certain formations. I can have an alpha helix, or I can have a beta pleated sheet. And then anytime we talk about them, we can always draw a picture to show what they look like. If you're a person of words, you can just describe it with words. But to me, I can throw up a picture a little bit quicker than I can type out like several sentences, especially if I'm handwriting. Typing, I'm pretty quick, but handwriting, I'm kind of slow. So the other thing that's significant is what holds their shape? Hydrogen bonds. So I don't care if you're on the AP Bio test or if you're at Texas A&M University and they're asking about levels of protein structure, it's important to them to know which bonds are available. So secondary structure, yes, these are held together with covalent bonds. Specifically, they call them peptide bonds. Amino acids held together are held together with peptide bonds. It gets its own vocab word for some reason. But as far as like what holds the shape of this sort of coil or this little folded thing right here, the beta pleated sheet, it's hydrogen bonds. Whenever I heat up a protein too hot, like we were saying before over here, it's these hydrogen bonds that break that make my protein denature first. So it's not that I'm breaking these bonds. That takes like an oven or something. Now it's like it goes from chicken muscle to like charred stuff. But when I just denature from like a fever, I'm breaking these hydrogen bonds in here. The third level is when I start taking several of these and joining them. So maybe I've got an alpha helix, a beta pleated sheet, another beta pleated sheet, another alpha helix, and they start bonding with each other. So maybe they form some hydrogen bonds. Maybe they form some covalent bonds. All bonds are fair game. Maybe this guy's got a full-on plus, and this guy's got a full-on minus charge. That'd be ionic. And then even those things that were forces, do you remember what those were called? There was, yeah, intermolecular forces. There was an intermolecular force. One intermolecular force is a hydrogen bond. The other one was like a weird kind of German sounding name. Van der Waals. And I don't know if Van der Waals really is German, but whenever I think of like V's and W's and stuff like that, I think of German. So Van der Waals forces. Van der Waals. And then quaternary. What's the difference between like just a bigger tertiary and a quaternary? Like, is it just once I add enough, like once I add four or more or five or more alpha helices and beta pleated sheets, or like what do I have to do to make quaternary? Huh? Yeah, now I need a second or a third or multiple chains. So right here, this is just one big long chain, one piece of ribbon, so to speak. But then if I have a second one and I add it in like this, and they start bonding with each other, all bond types are possible. So all four bond types are possible. Then now I have quaternary structure. Why is this important? Because shape matters. Shape determines what this enzyme looks like, and that determines what substrate it fits. So I get why shape matters. I don't quite understand why they're so picky and so obsessed of all the stuff we could teach you in biology, why we break it down by like zooming out and looking at the whole protein and then zooming in to where we're eventually looking at individual amino acids. I don't know why it's such a big deal, but it is universally in all biology professors' minds. It's a big deal. So it just is. So I've given the analogy before, but knowing this is kind of like knowing how to properly do a works cited page. Whatever you put on your works cited page, I bet I could figure out where you got your source from. But an English teacher will have none of that. You put a semicolon there, and that is supposed to be a comma, so it's wrong. And you're like, why? And she's like, I don't know, but it's wrong. You're like, okay. All right. So I've got another question for you. If you have that document pulled up, then it's going to be like right after the last one we did. So it starts off on page 1 here. And on the top of page 2 is a question from 2009, number 2. So back in the day, this was a 10-point question. Okay, so it says, number 2, ATP and GTP are primary sources of energy for biochemical reactions. You're like, all right, I agree. Part A, describe the structure of ATP or GTP. Part B is going to have you explain how chemiosmosis produces ATP. So we'll focus on A and B. I don't know that we'll get to C and D today. So our goal is to look at A and maybe get into B a little bit. So how would I describe the structure of ATP or GTP? All right, give it a shot. Describe the structure of ATP or GTP. And then B says explain how chemiosmosis produces ATP. Yeah, that's better. Nice job. I'm looking in the chat. Cool. We'll add on a little bit when you're mentioning the phosphates connected. We'll look at a little bit of like why phosphate? Like why does that matter? All right, let's look at part A first. We'll come back to part B. So the structure of ATP or GTP. So it turns out there's ATP, GTP, CTP, UTP. Just like in RNA, there's A's, U's, G's, and C's. Well, there's a TP version of all of those. So any of them could work as a good energy source. For whatever reason in nature, ATP wins out. So there's a few places where our body uses GTP instead. You don't have to know these, but just to kind of make a point, when ribosomes are like moving along, when they're reading the mRNA and doing translation, those ribosomes actually run on GTP instead of ATP. You can kind of think of it how, you know, cars, most cars run on gasoline. That's kind of like ATP, but some run purely on electricity. Or most things around here are running on electricity, but they could run on some other type of energy, something like that. So the structure of ATP and the structure of GTP is identical, except one of them has a guanine in there and one of them has an adenine. But that's not the part that matters. Which part of ATP actually matters? Is it the adenosine part? No, it's the triphosphate part. So if all you did for your drawing was this, let me switch cameras. So for your drawing, you had something like this. Ignore my mitochondria. That's going to be for part B. But if you had something like adenosine, and then I have phosphates, that'd be fine for my drawing. They don't expect you to know that an adenosine is a ribose molecule joined to, joined to an adenine molecule. All right, so I've got these phosphates. What's special about the phosphates? Like, why is ATP the energy source for pretty much everything? Yeah, ATP is highly unstable. What's unstable about it? Anytime I see phosphates, the first thing I think of, if somebody's like, oh, well, there's a phosphate, the first thing I think in my brain is, oh, well, if there's a phosphate, then this is true. Do what? Oh, go ahead. Go ahead, Aiden. Sorry. Yeah, they're negative. So anytime I see phosphates, I think of like, oh, they're negative, and that means they're polar. So if I'm talking about phospholipid bilayer, those phosphate heads are negative. That means they're polar, and that matters. If I'm talking about ATP, these have negative charges on them, and that matters. For reasons that don't really matter to us right now, the ones in the middle have a negative charge each, and the one on the end has two negative charges. Doesn't really matter. The important part is that negatives bonded to negatives bonded to negatives. So as Mercy was saying, that means they're highly unstable. When negative things are next to negative things, they want to repel. But these are bonded to each other. So this guy and this guy and this guy, they're all like repelling apart from each other. At the beginning of the year, I'd say like, this is phosphate. Maybe this is phosphate number 2. And this right here is phosphate number 3. And this is the bond holding them. So it's like they're repelling each other. So what's significant is in ATP, there's 3 phosphates. Phosphates are negative. When I have negative things bonded to negative things, that's unstable. And so if I want to break off a phosphate here or here to make like ADP, or even AMP, monophosphate, if I want to break off a phosphate, it doesn't take much energy because they're already repelling each other, and then it releases a ton. That means it's a good energy source. Most good energy sources take very little to get them going, and then they release a ton of energy. If I've got a gallon of gasoline, it takes a spark of energy to get it going, and then it releases enough energy to like burn the house down. So most things, batteries, they're highly unstable, right? They start oozing crap and everything else. So most of our good energy sources are highly unstable: kerosene, gasoline, um, batteries, and it takes very little energy to break them. And then when they do, they release a ton. If I was drawing a graph, it takes very little activation energy to break off a phosphate, teeny tiny bump, and then they release a ton of energy when they do. So that's why. So what's special about ATP or GTP? It's the phosphates. I've got three negative things stuck to each other. So those bonds are unstable, easy to break, release a ton of energy. That's why. Speaking of ATP, part B said, explain how chemiosmosis makes ATP. So first of all, what's chemiosmosis? If I don't know that, I'm kind of screwed. Huh? Yeah, it's the proton pumps and ATP synthase, kind of this part of the mitochondria. That process is called chemiosmosis. So take maybe 30 more seconds just in case you didn't get a chance. Jot down a few things about chemiosmosis. We'll walk through it. And here in about 5 minutes, I think it'll be TCTV time. That's a great question. So the question was, what's oxidative phosphorylation? So they're one and the same, but it's sort of like whose point of view you're looking at. So think of it this way. Picture football. You might not be a huge football fan, but you still know like the quarterback throws it and the receiver catches it. So I watch a play. From the quarterback's point of view, what did I do? I threw the ball. From the receiver's point of view, what did I do? I caught the ball. So what happened? Was it throwing? Yes. Was it catching? Yes. They're both right. So oxidative phosphorylation is if we follow from the point of view of oxygen. So like powering those proton pumps with oxygen, that's oxidative phosphorylation. When proton pumps work with an ATP synthase to make ATP, they call that chemiosmosis. However, anytime I've ever seen a question and they're like, explain oxidative phosphorylation, they also explain chemiosmosis. So if you're ever talking about one, go ahead and talk about all of it. Technically, oxidative phosphorylation doesn't involve the ATP synthase. It's just powering the proton pumps. They pump in the protons, everything's ready to go. Okay, cool. Now, chemiosmosis is these protons flow through the ATP synthase and make ATP. So yeah, good question. And it's super annoying because you'll even see books where they talk about how they're different, but then when they ask you questions, you answer both. So it's kind of annoying. All right, so over here, my folded inner membrane. What was that called? Do you remember? The inner membrane of the mitochondria was all folded up, and the name did not make sense. It wasn't like endocytosis, you bring into the cell, that's logical, or passive transport is no energy. It's called cristae. Again, I don't know why. Crist means cross, and I don't see one. So this inner membrane is folded. Why is it folded, though? That's the most important part, probably. Yeah, good. I can increase how much surface area I have. So if there's something important going on this inner surface, the more surface I have, the more of that crap I can do. So on this surface, I have our ATP synthase. And I've also got proton pumps. And they'll be in the ratio of like 3 to 1. I should have 3 pumps for my ATP synthase. All right. So we'd always mentioned that ATP synthase has 2 important characteristics. Can somebody give me one of those? I'm going to try to get somebody that hasn't answered yet. It can be somebody online also. So what's one characteristic of ATP synthase? Nice, Alice. Good. It's hollow and it spins. So those are our 2. This is hollow and it spins. Why do I care that it spins? I mean, it's surprising that I have little spinning motors in my body, but what's it accomplishing when it spins? Yeah, I'm taking an ADP and I smash a phosphate onto it. So as it spins, it's holding an ADP and a phosphate, and it's kind of like it's going like this. It's like a machine that spins and smashes them together to make ATP. So that's why we need it to spin. Why does it matter that it's hollow? Why would I emphasize that? Like, of all the crap to point out, like, okay, I see why spinning matters. Why does hollowness matter? Huh? Yeah, protons pass through it. And if protons pass through this way, in my picture, right to left, we'll name these sections in just a second. But if they pass through this way, then it slams together an ADP and a phosphate. If the protons pass through the other way, then the machine runs in reverse and it rips them apart. I want to make ATP. It's ATP synthase. So I want protons to always flow that way. By the way, what is this area in here called? It's a space between two membranes. Intermembrane space. And then what was this area out here called? Where like the Krebs cycle happens and stuff like that. Yep, matrix. I see Christine online got it, and then somebody said it, Luke said it here in class, the matrix. So technically, I'd say I want protons to move from the intermembrane space out into the matrix. If they go the opposite way, matrix to intermembrane space, now we rip apart ATP. Okay, are these protons like is this more like a pump, or is it more like a channel, or like is it active, is it passive? What can you tell me about when this is moving? What can you tell me about it? Is it active or passive? It's passive. So that's just regular old diffusion, right? So diffusion, they move from high to low concentration. There's a little bit more to it though. They now that we've done the nervous system, when charged particles move, isn't that current? Like, isn't that electricity? So we can actually measure the voltage across here. When we want to see how fast the mitochondria is working, we can measure the voltage. As protons are flying through there, the voltmeter is like moving. In fact, that's one of the first ways that they confirmed this was actually happening. At one point, scientists were like, hey, I think protons flow through there. And they're like, how are you going to prove that? And he's like, well, moving protons means voltage. So let's hook up a voltmeter. And all of a sudden, they could sense the protons flying through there. So they call it an electrochemical gradient. It's not just a chemical gradient, high to low. It's also an electrochemical gradient. So it means it's a little bit more complicated. Not only are they moving from high to low, but they're also kind of like repelling each other. Those pluses are kind of packed together. They're kind of repelling each other out. Okay, so that works great until we reach equilibrium. How do I make sure that I don't reach equilibrium? How do I make sure that there's always more protons in the intermembrane space? Right as the AC kicks off. Yep, proton pumps. And so they're going to take the protons that went out here, shove them back inside. So we're just using the same protons over and over again. That's a proton pump. A proton pump. What do I know about it if it's a pump? Active transport. And what's usually my energy source for active transport? No, go ahead. In this, I'm going to need an alternate. What do I usually use? Anytime I need to use energy, what chemical do I usually use? Yeah, I usually use ATP, but now I want to use something different. So I want to pair these with something different because I'm trying to make ATP. So if possible, I'd like to not burn some in the process. If I needed to, fine, but so what do I power them with? They pump protons, but I power them with electrons. So electrons is what's going to power these pumps. So what students find confusing is they're called proton pumps. So what students find confusing is it's like, well, they're proton pumps. So what do they pump? And you'd be like, well, protons. But what powers them? Electrons. So we usually, not always, but we usually name something based off of what it does, not its energy source. My lawnmower mows the lawn, but it runs on gasoline. My weed eater eats weeds, but it runs on gasoline. I don't call my car the gasoline powered thing. I don't call my mower the gasoline powered thing. I name it based off of what it does. So proton pumps are named off of what they do. They pump protons, but their energy source, electrons. And then the last question, where did I get those electrons from? There's a couple correct answers. I guess three. Where did the electrons come from that power those pumps? By the way, I see you, Davey. You got that right.
something.
Yeah. So I've got my two electron carriers from like the Krebs cycle. So there's NAD. When it's full of electrons, it's NADH. That's a good answer. What was my other electron carrier? Yeah, FAD. When it's full, it's called FADH2. So those are good answers. Or if I go far enough back, you could say like, well, originally it's like food. You're sort of stealing electrons from food, and that's what's powering them. So those are like the three answers that I think are good: NAD, FAD, or food. All right, it's a whole lot of review all at once. Thanks for hanging in there. We are done for today. If you're on the Zoom and you have questions, stay on. The two of you guys that wanted me to look at your quiz, I will, and I'll send you an email to let you know the outcome of that. And otherwise, see ya.